about respiratory anatomy and how some of that anatomy is tied to very specific functions of the respiratory system. So to start, we have this figure in the upper left here, which is a cast of cadaver lungs. What that means is that they take the lungs of a cadaver and they fill it with a solution that solidifies. And so what it does is it allows you to see the branching structure of the respiratory tree. And so at the very top here, it's sort of cut off. This would be the trachea. And I want to talk a little bit about the respiratory tract above the trachea as well. Not too much crazy detail, but it is important to understand some of the functional roles of the respiratory tract of the trachea and above. It's called the upper respiratory tract. And really, if you think about it, right, air can reach the trachea either through your mouth, right, and then through the pharynx, of course, the pharynx being your throat, and then at the bottom of your throat, your pharynx, it would split between going to your esophagus and your GI tract, or air going to the respiratory tract. But air can also go through the nasal passages, right, in the nasal cavity. And the key component to talk about there would be the lining, right, the mucous membrane that lines the nasal cavity. And then as well, once we get below the pharynx, right, because remember that's dual air, and food and drink passageway. Once we get past the pharynx and we're in the trachea where it's only air again, unless of course you laugh while you're eating or something, right? We're going to go back to that mucous membrane, right, or a very specific type of that mucous membrane. So the the nasal cavity has this specific mucous membrane, and then the trachea, and then some of the branches of the trachea off of the trachea. So the primary bronchi are here, and then if it keeps branching, and then you're into the tertiary and then quaternary beyond that, right, smaller and smaller. And down a significant number of those branches, we have this mucous membrane, and it has, right, so upper respiratory tract, I'm going to put this as generically upper, lined with, it's a pseudostratified columnar epithelium. I'm going to abbreviate that. I'm going to abbreviate it as PCCE. Pseudostratified means it looks like it's more than one layer, but it's one layer. Ciliated, I didn't say that term. Ciliated means it has cilia on its surface, and cilia serve to move the mucus that this membranous layer produces. We'll come back to that. Columnar, right? The shape of the cells are tall and thin. And of course, it's an epithelial tissue. So pseudostratified ciliated columnar epithelium lines this upper respiratory tract. Again, the nasal cavity, and then the trachea and larger branches of that come off of the trachea are lined with this mucous membrane, a component of which is pseudostratified ciliated columnar epithelium. Okay, what's the role here? What's the role of this membrane? Right? Well, it produces mucus. The two key things are it produces mucus and it has cilia. And so the role is protection. You might say, well, protection from what? Well, the mucus serves to trap airborne particulate matter, right? The dust and dander and pollen and bacterial spores and fungal spores and all sorts of things that are suspended in the air we breathe. Much of it gets trapped in that mucus layer. And that mucus layer is being moved by the first C here, right? By the cilia. And it's moving that mucus toward the back of the pharynx. So in other words, in the trachea, the cilia would be sweeping upward, right? They would be moving the mucus upward out of this blind-ended respiratory tract, right? It's not a flow-through system. We breathe air into these spaces and then we breathe it back out. And so any particulate matter that gets trapped down here has to be moved back up the pipes, right? So the cilia would be sweeping this mucus upward. The cilia in the nasal cavity actually sweep backwards towards the throat. And maybe you're getting the gist of this, right? So we're sweeping this mucus with all the trapped particulate matter towards the throat. Well, right, up from the lungs, up through the trachea, or backwards through the nasal cavity, and it ends up in the back of your throat. So guess what we do with it? We swallow it, right? And that is a protective mechanism because it takes all that particulate matter that ends up in our respiratory tract and it puts it into our GI tract where it gets, digested by hydrochloric acid and enzymes, and it ends up not causing us any harm, right? If we were to allow it to remain in the respiratory tract, especially things like the bacteria or bacterial spores at least, and things like that, you would set up infections quite readily. And people with ciliary disorders where the cilia don't work properly often end up with more respiratory tract infections than normal. Okay, and that's actually called a muco, and I know we're combining function and anatomy here, but that's called mucociliary defense. Right? Muco for the mucus, and then ciliary for the cilia that are moving that mucus, and it's a defense system largely protecting the lungs from that particulate matter. Okay, now let's talk about what happens as we move out this respiratory tree, right? The farther we move out these branches, remember these are branching and branching and branching, right? The branches are branching, right? And it keeps branching. And, all the way out here, and you start to get to these peripheral regions out here. I could circle anywhere, but these peripheral regions. Now we're looking at this image, right? If I were to take this little spot, draw an arrow. Now we're looking at this image, right? Where we're talking about, it's called the terminal lung. There's actually a model of this in the lab. You might see images of it if you're taking the lab course right now. And what we want to look at here is how the structure of the lung differs from this sort of, and one of the things I didn't point out, in the wall up here, not only is it lined by a mucus layer, but in the bigger pipes, it's got cartilaginous rings. So we form these sort of rigid pipes. They don't constrict or dilate, and they're lined by this mucous membrane that's protective. But when you get down to the bottom, the cartilage is gone. We end up replacing it with this red stuff here, which is this red layer, which is smooth muscle, right? And the small pipes are called bronchioles. Now notice we have blood flow here, right? Notice, that the artery, right, to remind you of something else, this is an artery, but it's blue, which means the blood flow is going this way, right? It's not coming back from the alveolar surface, it's going to it, and it's blue because it's deoxygenated, right? And so it's going to the lung surface down here, the alveolar surfaces, or the alveoli, right, to get the oxygen and return as oxygenated blood in the pulmonary veins. Okay, again, we're getting a little ahead of ourselves here. What do we really want to focus on besides, those are important elements, but we want to focus on this sort of branching, right? There are actually, and you don't need to know the number, but there are actually 23 degrees of branching from the trachea to the alveoli down here. And what that does is it provides about 300 million of these little air sacs. And again, I don't care that you know the number, but it's an enormous number of these little air sacs. Why? Because of Fick's law, right? Fick's law explains the factors which maximize or minimize diffusion, however you want to look at it. And surface area is one of them, right? And so having so many of these little tiny alveoli increases the total surface area of the lung. It's kind of like saying, well, a box the size of a sponge has the surface area of just the outside of the box, right? Whereas the sponge, if you measured the surface area of each of the little bubbles within a sponge and added those all together, exact same size as the box in terms of total volume, but the surface area of that sponge would be 10 or 100 or 1,000 times higher, depending on how big the bubbles are, right? Well, these are really small bubbles, if you will, right? The little tiny air sacs at the end of the lung. All right. We notice that we have smooth muscle. That is going to allow us to, and again, at this terminal lung, at the distal branches of this respiratory tree. And what that's going to allow us to do is shunt the movement of air within regions of the lung. Local control, right? Remember when we talked about homeostasis, there's always this first level of control at the target, the microscopic tissue. And having smooth muscle here allows us to direct airflow to where there is good blood flow and vice versa. We're going to come back to that later. It's called ventilation perfusion matching, right? Ventilation means air movement. Perfusion means blood flow. And we want the two to sort of match, right? You want to send blood flow where you have air and vice versa, right? Within the regions of the lung. Okay. Another thing I want to point out is the capillary beds, right, coming off of this pulmonary artery, form a network of alveolar capillaries. I'm pointing here, right? Our capillary beds that literally coat the surface of those alveoli. And by the way, the alveoli are always drawn as spheres, and they're not spheres. They're actually what's called a polyhedron. They're flat-sided, but whatever. So they look a bit more like a soccer ball that's not blown up all the way, right? Where the panels would look kind of flat. That's what the alveoli look like. But they're always drawn as spheres, and that's okay. But those spheres, or polyhedrons more accurately, are covered, just literally covered in capillaries. And again, that comes back to the Fick's law, right? If we have a whole, this enormous surface area of alveoli, but you didn't have the matching capillary surface area to sit on top of it, there'd be no point in having all the alveoli, right? The two need to go very close hand in hand so that our large surface area of, for air distribution, if you will, to a large surface area, it needs to be matched by a very large blood surface area. In other words, the blood has to cover the blood flow has to cover the surface of the alveoli. And of course, it's contained within the capillaries, but those capillaries are really close together. It's actually called sheet flow because the capillaries almost merge, right? And they form what looks more like a blanket of vessel or a sandwich, if you will, of blood flow between two layers. It forms a very large surface area for blood flow adjacent to these alveoli, right? And of course, we want them to be very close together because also in Fick's law is distance, delta x, right? We want the two to be close together to maximize diffusion. Okay, so upper respiratory tract, we had the mucociliary defense, right? The mucus production, the cilia moving that mucus. When we get to the lower portions, we have this control of, we didn't talk about it, but we have control of blood flow, and we have control of the bronchioles in terms of the smooth muscle here, so we can shunt airflow as well as blood flow. And then once we get down to the alveoli themselves, we have this really large surface area of alveoli matched by a really large surface area of capillaries. Now note, before we take a closer look at those alveoli, The outside of the lung is surrounded by this pleural membrane. I'm going to bracket these things right here. This is what hopefully you learned in A&P 1 as a serous membrane. And it serves a couple of purposes, right? So a serous membrane is always two layered, right? There's the visceral layer, which is like shrink wrapped onto the organ. So that's stuck right to the lung surface, right? Here's the lung, here's this layer. Then there's an outer layer, the parietal layer, that's sort of stuck to the inside of the container that the lung is in, or there are other serous membranes like in the GI tract. But so in this case, the parietal pleura meaning the serous membrane wrapped around the lungs, the parietal layer would be shrink wrapped onto the inside of the rib cage or to the top of the diaphragm. The two are separated by this space called the pleural cavity that has pleural fluid in it. And again, in terms of sort of foreshadowing some function later on when we talk about ventilation, the fluid is there to help the lungs stick to the thoracic wall. And we'll find that that's really important to the function of breathing. We have to have this fluid layer between the lung and this parietal layer, which is on the inside of the rib cage and the diaphragm, so that the two stick together. You don't want your lungs becoming separated from the thoracic cavity wall, right? That is really the job of this pleural membrane. And again, with its two layers, the visceral inner and the parietal, the outer layers. Okay, we'll come back to that in terms of functionality later on. Now, what I want to do, though, is talk about just a little bit about one more key anatomical structure. And I'm going to sketch it because I'm just going to blow up an alveolus here. So the alveoli come in sort of clumps, as you can see over there on the right. I'm only going to draw one here, even though that's not anatomically sort of how they exist. I'll make it a little bit bigger, right? And there would be others, like it'd have some branch over here to another one, right? Sort of sitting adjacent to it and so on. And they are formed of, right? Remember we talked about the epithelium of the upper tract, and it was here it is, right? Here's the epithelial layer of the upper tract, is this pseudo-stratified, ciliated columnar epithelium, right? And it served its purpose. Well, when you get down to the bottom and you get to the alveoli and the last couple branches of the bronchioles, right? I'm not going to draw this in the other guy. We end up with a label on here, squamous epithelium. And to be even more accurate, it is a simple squamous epithelium. Simple means single layered. Squamous means flat, right? And so these are a single layer of cells and they are very flat. We are back to thick, right? Thick. Because we have to get oxygen from in this air into the blood, which I didn't draw yet, right? But I'm going to draw the arrow already, right? Into the blood that's going to be adjacent in these capillaries that are going to sit right on top of it. And again, the capillaries are made of, guess what? Simple squamous epithelium. Right? And I said these capillaries form this sort of almost continuous layer, where they're just one right next to the other. Again, I won't draw all of them, but you get the idea. Right? These are the cells of the wall I'm drawing. Right? You could squeeze some nuclei in there if it helps. You get to recall that. Right? And so the last thing to identify is a structure, really, to identify right here in the respiratory tract is this. Right? Within that box is the respiratory membrane. Right? And so it is the alveolar wall plus the capillary wall, and if we wanted to try and squeeze it in here, there's a little skinny Basement membrane. Remember, that's that extracellular matrix layer, right? Little skinny layer of extracellular matrix that kind of sits in between everything here, right? And so, right, so the respiratory membrane equals the squamous layer of the alveoli, which I don't have room to write all this down, but the squamous layer of the alveoli in blue, the squamous layer of the capillary wall in red, and the shared basement membrane in between the two, right? And the key thing to remember here is delta x, right? Fick's law, right? Delta x is the distance. We want to minimize the distance that our gases need to diffuse. And I can put a different color. I can put my carbon dioxide diffusing in just the exact opposite direction. And these gases are lipophilic, which means they don't care about going between cells. They diffuse right through lipid bilayers. Not quite equally, but they both are lipophilic, and so we don't really have to care where we draw our lines, our arrows, because going through membranes is just fine for these gases. Okay? Now, the alveoli are made of these, right? This is called a type 1 alveolar cell, if you want to get specific about it, right? This is a type 1 cell. But there's a type 2 cell. And the type 2 cell sort of sits in between things here, right? And I'll just sort of highlight this guy a little bit. There are multiple, more than one of them. There's not one per alveoli or anything like that. But this other cell type sort of sits here along the wall, or embedded within the wall. And the function of that cell type, that's a type 2, by the way. Get rid of this other guy. Right, so that's a type 2 cell, and it punched a hole in my alveolus there. This guy's type 2. And its job is to produce this substance called surfactant. And again, I don't want to get into all the details of surfactant, but it's there to help keep the smallest bronchioles, probably not so much the alveoli, but the smallest bronchioles from collapsing. So again, that's going to be really important in terms of function and providing what's called lung compliance. In other words, an ease with which air can sort of move in and move out of the lungs. Right? And so these type 2 guys, again, running down a room over here, produce this stuff called surfactant. Surfactant. And surfactant is really, that's a T over there. Surfactant is like a detergent. And what it's going to do is sort of reduce the surface tension in there. And again, we'll come back to that and talk to it when we get more about the functionality of ventilation and the ease of air movement. Okay? So, down here at the distal lung, we've got our ability to shunt through the smooth muscle. We get down to our alveoli, or individual alveoli, of which we have a huge number, right? Remember, don't forget, huge number to provide large surface area, right? That's in Fick's law. We mentioned that pleural layer to aid ventilation, again, for surface tension. We got surface tension in 2 places. We're going to have to be very careful about that, right? One where we want it and one where we don't. Again, clarify that later. And then when we get down to the alveoli, we want to focus on the key anatomical structure here, which is the respiratory membrane, which is the separation between air and blood, right? It is delta X. It's the distance that you need to move things to get from air to blood, or vice versa, right? We'll come back to the type 2 cells that produce surfactant, and there's actually a third type of cell. I'll make this guy look a little bit funky here, right? A third type of cell, it's often called a dust cell, and this is a white blood cell type that actually serves to sort of like a janitor. It's sort of cleaning up any of the small particulate matter, right? If we threw some, that made it past the mucus escalator, like little tiny particles of dust and dirt that might make it down here, well, this guy's going to gobble them up. It's called phagocytosis. It's going to gobble up those elements of dirt and dust that make it to the really small fragments of dust in the air that make it to the alveoli so that they don't accumulate there. Okay. The last thing I want to do is point out the Weibull model, and that's just really generically sort of summarize some things. Let me do this. So this is the Weibull model, a little bit bigger. And the Weibull model is basically looking, right? So here's our respiratory. So here's the trachea right here. Right, there's trachea. Right, there's division 1 of the, or 0, of the respiratory tree. And then you see it just starts to branch and branch and branch as we go. It's sort of like this person's laying on their side, right? And the branching and branching occurs, and you have what's called the very, up to branch 16. Look at this, in the bronchioles. So this is a lot of branch. These things are tiny. You're talking about bronchioles that are probably a fraction of a millimeter in diameter. And that's called the conducting zone, because its job is simply to conduct air down to and back from the respiratory zone. Right? In other words, there is no gas exchange in that area. It's actually called anatomical dead space because it doesn't serve the purpose of gas exchange. It's just a really complex snorkel, if you will, to get to the gas, oxygen, the air to the respiratory zone. And then, of course, the exhaled carbon dioxide out of that zone. Right? So the respiratory zone is everything past that, 16th branch, if you will. Right? And again, numbers are irrelevant, other than know there's a lot of them. And what you'll notice is that, see these little guys? Right? All of a sudden, oh, look, they start to show up. Those are alveoli actually sticking to the walls of these, very distal bronchioles. And then they become even more numerous until you get to the very end, right? And the 23rd division of the bronchiolar tree is the alveoli, where it's just alveoli. There's no more branching, right? You're there. You're at the alveoli. And the key thing to look at here is this guy down here. Look at this. This is cross-sectional area. And again, getting right back to Fick's law, right? So we want to run our air down to the exchange surface through a really large pipe, or series of relatively large pipes, right? But again, it's not about gas exchange there. So the surface area is actually relatively small. And then the branching and the presence of the alveoli, watch what happens to the surface area, right? The closer and closer we get to the end, whoosh, right? Off it goes. Huge surface area at the bottom to maximize our gas exchange. You'll notice this next segment here just sort of points out some of the key structures, which again should already make sense. Look at the epithelium, right? Up at the top, it's that pseudostratified columnar, right? Labeled right here as columnar. And then it sort of like shrinks in height and it loses its cilia. And then down at the alveoli, it is simple squamous. Look at the cilia. Do we need cilia way down here? Right? Do you need cilia in the alveoli? You better not, because if you're making mucus to move cilia in the alveoli, what did you just do to your diffusional distance? Right? We do not want to be diffusing through a thick layer of mucus to move those gases. It's actually exceptionally dangerous. So we don't want cilia, we don't want mucus down at the distal lung, at the respiratory zone. So the cilia go away. The mucus secretion, right here, the cilia, those two are going to go together. Right? They're going to disappear at about the same time. And of course, we lose the cartilage because we don't have to hold open those big pipes once we get down to the bottom. We actually want to be able to constrict or relax them so that we can shunt air to and fro amongst the regions of the lung. Okay, and again, that's called the Weibull model. But that's not really that important. But he was the one that sort of laid out this model of respiratory anatomy and how it ties to the function and the really large surface area with our 300 million alveoli providing it. All right, I think we're good.